Welcome to the FarmBits podcast, a product of Nebraska Extension Digital Agriculture. I'm Jackson Stansel.
And I'm Samantha Teaton, and we come to you each week to discuss the trends, the realities, and the value of digital agriculture.
Through interviews and panels with experts, producers, and innovators from all sectors of digital technology, we hope that you step away from each episode with new practical knowledge of digital agriculture technology. Hello and welcome to the FarmBits podcast. This episode marks the beginning of our precision crop protection series, in which we will investigate the technologies that are starting to revolutionize the way that we approach crop protection. It is also the first time that we will have a new co-host joining me here on the FarmBits podcast. Zach Rystrom is a Doctor of Plant Health student here at the University of Nebraska and is interning with the On-Farm Research Network for the summer. Welcome, Zach, and we're really glad to have you here on the podcast.
Thanks, glad to be here.
Awesome. On this episode of the podcast, we are welcoming Dr. Erik Christian, Agronomic Services Manager at Trace Genomics.
Trace Genomics is enabling greater insights about the biology and chemistry that interacts within soil to drive forward management decisions tailored to the particular potential and risk within a given field.
Dr. Christian discusses Trace Genomics, how they're revolutionizing the way that growers approach crop protection, and how they're seeking to be an enabling data set for advanced analytics and insights in agriculture.
There's a lot of interesting content in this episode, so let's get right to our interview.
You guys do good work, so I'm glad you're documenting all this. You're talking to some really cool people, so excluding me, but it's pretty cool. So I'm glad. Thanks for the opportunity, guys.
Absolutely, no, that's been the best part of the experience for me. It's just been learning a lot of new things. Sam and I talk about that a lot. That we've almost learned more from our podcast interviews than we have, from just being in classes or doing just about anything else. I think probably talking to farmers is the only other place we learn more. But
yeah, well, networking. I mean, you've talked to some really influential people. So I am in Ames, Iowa right now, but I'm originally from Central Iowa, so I'm not too far from where our lab is at. And I grew up on a farm where we raised corn, soybeans, a bit of hay, but we also raised livestock. We had pigs and cattle and those sort of things. But from there, I went to Iowa State, got a degree in agronomy, and then ended up going on to graduate school. And my area of focus in graduate school is on seed physiology. So really focused on germination, dormancy, things like that in seeds. I spent a bit of time in the seed industry as an agronomist as well, and really enjoyed that, getting to work with growers to really help them find the right variety, right hybrid. But then, as we talked earlier, I stayed on at Iowa State actually after my graduate career and did a bit of teaching, really focusing on undergraduate teaching in the areas of crop production and things like that. And was lucky enough to get to come to Winfield United after my period of teaching, and really worked in the area of soil fertility and plant nutrition. Winfield United at the time had two soil testing labs, one here in Ames and one in Indianapolis, Indiana. So I had a counterpart in Indiana, and we really helped support the agronomists as salespeople when it came to soil testing, soil fertility, and things like that. But then came to Trace Genomics in January 2019. And my main functions at Trace Genomics are really two things. One is customer support. As I mentioned, we do a lot of work with our customers from the very beginning of understanding our technology to designing the soil sampling, to, on the other end, really helping them interpret the data and to drive that decision making. But I also lead our applied research and development group, where we're developing those deeper insights, where we're creating models, where we're looking at the larger data set to understand what can we learn from this to really help folks out. So
seems like a pretty important role, and you've seen a lot of different sides of the industry over the course of your career.
So one interesting thing. So again, I didn't really ever envision my life in going to a startup. You just never know where it's going to take you. But the interesting thing about Trace Genomics, we actually, starting in California, we worked a lot in the specialty crop area. A lot of lettuce, grapes, almonds, crops like that. And so in any given day, I was on a call earlier today talking to some lettuce pathologists and different things like that. Getting to work with a lot of the non-corn and soybean areas of the United States has been really, really interesting. We work with some international folks as well. And so to me, being an Iowan, an agronomy graduate at Iowa State, we learned a lot about corn, soybean, because that's sort of what most people were going to do. But it's been really, really exciting to branch out, to work with people across the country and in the world in different crops. When I taught my sophomore level crop production course at Iowa State, it was really focused on corn, soybeans, some forages and small grains. But one of the things that I'm sort of living out was I wanted the students to really understand the principles of crop production, because you don't know what you get that big corporate job out of college and they're going to send you to Alabama, right?
Right. Yeah.
And what are the kind of things you're going to run into there? You may be looking at corn, soybeans, but you may be also looking at a lot of other crops, and being able to understand some of the fundamental principles of crop production and applying it. Sure, there are nuances even within corn, soybean production. Getting into perennial crops, getting into irrigated crops, but all plants really need a lot of the same things, right? And so if we can make sure that we're getting those plants what they need, that's the key. So to me, that's been one of the most exciting things about Trace Genomics, is just getting to work with a lot of really interesting people, and a startup. I mean, we move fast. And we can really change quickly. Large companies, you focus on this really narrow piece of what the company does. You're going to be sticking with that for a while. And to be honest, can you really have a huge impact on the direction of the company? Unless you're one of the decision makers, you're going to sort of deal with what you have, right?
Right.
Yeah. So I was based in this lab. So before Trace Genomics, I worked for Winfield United.
Okay.
And I was based out of this lab and got the opportunity to go to Trace Genomics. And at the time, in 2019, our main offices and labs were in San Francisco. Okay. And again, so here we are sending thousands and thousands of pounds of Iowa, Nebraska, Minnesota soil to San Francisco. And it's maybe not the greatest idea. And anyway, we got the opportunity to purchase Solum in March of 2020. And so we did. So we really merged the two together, ended up closing our office and labs down in San Francisco, moved all of the lab processing here. Most of the folks that worked in our facilities in California just went remote. So they're all working from home. And now we send California samples to Iowa instead of sending Iowa soil to California. So it's pretty wild. There's one thing, there's a lot of things you learn about that. Number one, shipping soil is a costly thing to do. And
No joke.
we overnight the soil samples. So if you were going to say, take a soil sample and you were a ways away from Omaha and you were going to ship them to Midwest or whoever your favorite lab is, you put them in a box and you'd say ground ship them.
Sure. Yeah.
But since we're doing work and we're looking at the organisms in the soil sample, live organisms, we want soil samples to get to us as quickly as possible. So whether you're in, if you're in eastern Nebraska, you're in the Salinas Valley, California, you're in, in France, we typically, it's a little longer journey, but pretty much anywhere in the lower 48, we overnight all the soil to our lab in Ames. Wow. Which is expensive regardless of what you're shipping, but now we're talking about shipping soil samples. So, but anyway, I guess my last point there is what a lot of folks don't know is that there's a pretty large portion of the Southeast United States that's a fire ant quarantine zone.
I'm from one of those quarantine zones, Dothan, Alabama. Yeah.
Yeah. So if you were to go home and send soil to our lab in Ames, we have to take extra precautions because we don't want Ames, Iowa to become the epicenter of fire ants in the northern Midwest. Now, there's a lot of reasons why it probably wouldn't happen. We take that quarantine shipping very seriously, and so we make sure that folks follow those procedures. I guess maybe the one last thing I was saying when we cut off you've ever gotten a package from FedEx or UPS, you've seen perhaps the kind of damage they can do to
Oh yeah.
a box. And so if you think about that, and everybody packs a box of soil samples as full as you can, because you're like, this is soil, I'm not going to, I want to get my money's worth.
Yeah.
And you think about that, they drop it off the truck, they drive over with the airplane, they throw it here or there,
Yeah.
And now you got soil from Alabama that just got strewn all the way from there to Ames, Iowa. And so we get these 5-gallon buckets with screw-on lids, and then we zip tie them, no problem. And then when they get here, we have to follow a bunch of procedures where we're bleaching things and all kinds of, it's crazy.
You got autoclave all the soil when you get done and everything.
Yeah, you bet. All kinds of fun stuff. Even all the different, we surprisingly much different than most labs. We use a lot of pipette tips and different things like that. We have to autoclave all that, and so it's quite a to do. But we're a lab, and we work with people from across the world, and we got to make sure we're doing the best job we can to not spread that stuff.
Sure.
And if the USDA showed up and saw we weren't compliant, they'd shut us down, and that's not fun. So
yeah, as a quick aside before I let Zach get to the next question, I actually had to ship soil from Dothan, Alabama, the Headland experiment station that Auburn has down there, up to Harvard, which was where I was doing my undergrad. And so I did my thesis on kind of making a soil amendment. And I was trying to use some sandy loam soil because it was essentially trying to build something that would stop nitrate leaching. And I'm just like having flashbacks to all the issues that I had trying to ship something for, a kind of a non-certified lab. I had to have the USDA come in and certify the lab, make sure that all the proper procedures, make sure we had the right autoclave in the lab to get the temperatures we needed to. And it was a whole ordeal, and man, it was expensive to just get like 100 pounds of soil up there. It's like literally basically like a cubic yard of soil. That's about it. But golly.
Yeah, sounds like quite a struggle, but I'm glad you followed the rules. We'll get a lot of folks that'll say, our other lab doesn't ask us to do that. And you go, well, that's fine. I'm sure your other lab's great, but we gotta follow the rules. So, but yeah, getting from Alabama to Massachusetts, that's you gotta pay attention. So
yeah, that's for sure. Crazy.
Yeah. So what exactly is Trace Genomics, and what products and services does it offer to its customers?
Yeah, so Trace Genomics is an ag tech startup. It was founded in 2015 by two Stanford graduates. And really initially the company was focused on taking soil samples and extracting the DNA of organisms like fungi and bacteria, and then sequencing all that DNA and giving those results back to growers and agronomists, really to help drive more sustainable management. And so that's really the fundamental or foundational technology for Trace Genomics. But over time, we've really expanded. I can talk about that here a little bit more in a minute. But what do you do with that fungi and bacteria quantification? And so one of the main things we do is, as you can imagine, we quantify pathogens. So what we're doing, take your favorite fungi or bacterial pathogen, and what we're doing is quantifying the inoculum level in the soil. One thing we also do is, if you think about nutrient cycling, think about nitrogen cycling, for example, a fair bit of that is biologically driven. So if we're going to quantify the fungi and bacteria, we're also going to quantify those organisms involved in, let's say, the process of nitrification, for example, and quantify those organisms. And again, we're not, one distinction, we're quantifying the inoculum level, we're quantifying the organisms involved in nutrient cycling, but we're not quantifying the activity necessarily. That's certainly something that we're moving toward, but we want to be very clear that we're quantifying the amount of organisms in that soil sample. And then finally, something that we've added more recently is the quantification of a lot of the standard fertility parameters, pH, buffer pH, CEC, organic matter, and so on and so forth. So one of the things I think really differentiates us from traditional soil testing is not only the fact that we extract that DNA and sequence those organisms, but that we also have a data set that includes those chemical parameters, as we call them, pH and things like that. And we have a really fantastic data science group And we'll take all that data and we'll work with our customers to really understand the situation and to look at the interactions the best we can with the data we have today. So again, traditional soil testing lab, you take the samples, you drop them off there, and at the end of the processing, they give you some data.
Sure.
Our interaction with the customer starts much earlier than that. And this is actually one of the things that I do at Trace Genomics. I work with our customers to really design an appropriate sampling plan to help them collect the samples, to get them shipped to our lab here in Ames, Iowa. And then when the results are ready, I work with them to help interpret that data to help understand what it means. You know, if you get a phosphorus value back, let's say an Olsen phosphorus or Bray phosphorus value back, you can lean on 50, 60, 70 years of, you know, university research and correlation and calibration data to say, okay, I know exactly how much MAP or DAP I need to put on.
Sure.
When I give you a quantification of, let's say, Goss's wilt, you can't go to any researcher and say, well, what do I do with this value?
Yeah.
Right? So we have to work really closely with our customer to help interpret these values and to help drive those management decisions.
Sure. And one thing that you mentioned there, which I think is where we should probably start, because it's where everything starts, right, is the soil sampling. And so when you talk about putting together one of these soil sampling plans, and I guess coming at this from a precision ag point of view, I mean, we know there's a lot of spatial variability within a field, and I assume that there's a decent amount of spatial variability in terms of maybe different fungi or bacteria in the soil. And so how exactly do you design those soil sampling plans in terms of, you know, spatial locations, depths, and all that sort of stuff to provide you the information that you want to have to work with the grower?
Well, yeah, you're exactly right, Jackson. There is a lot of variability in a lot of soil parameters, not just the biological ones, but folks are not as used to sampling for the biological parameters. So really, we're just starting in this journey to understand what's going on in the soil biology. There's only a few companies that have this technology and that it's commercially available. So we're really all writing the book on this. And so one of the things that I like to start off with saying is like any company and any startup, we want to be disruptive, but we don't want to disrupt everything. So when it comes to sampling, I like to align sampling the best I can with what folks are already doing.
Gotcha.
I would say, you know, so if I came in and said, okay, I want you to sample in August in a cornfield in Nebraska, and I want you to take 80 cores per sample, and I want them to be to a depth of 2 inches, and you know, nobody's going to want to do this, right?
Right.
So we first start off the conversation with, what do you do? What do you do today? How's your standard fertility sampling? And start to get an understanding of what folks are doing today, and then make sure we align or modify that sampling to what they're looking for in the soil biology. If it's purely exploratory, well, then we sort of have to be, you know, we have to sort of aim for the middle. If we know we want to look for a specific organism, or we're trying to solve a specific problem, well, then we align it. But sort of one of the big things that doesn't change between what folks are used to now and what we're trying to do is the fact that when we get a sample, we want it to be a composite soil sample, so it has multiple cores that are distributed in an area. And we want that soil sample to be roughly 1 pound, 400 to 500 grams. So that looks no different. Our bags that we get in our lab here look no different than the bag of soil that you collect today. Now, the strategy of how we collect that, as you mentioned, the depth, you know, how many cores, where we're going to get that sample certainly can vary from what folks are used to. So even the depth, you know, you start looking into soil testing and soil fertility, the depth that we soil sample in Iowa is a different depth than they soil sample in Indiana.
Interesting.
And, you know, it really has to do with that correlation and calibration. It has to do with historically what folks, you know, deemed was the depth of incorporation of nutrients and lime and things like that. So, you know, talking to folks in Nebraska, you know, most often we're going to be collecting a sample that's probably 0 to 6 inches, which is pretty common. Probably when you're talking about a 6-inch sample, you're probably looking at 10 to 12 cores would make a nice sample. You know, folks that are really in a hurry and have thousands and thousands of acres to do typically will do less than 10 cores, but that's what we like to do. And, then you get into the grid versus zone. And that's a bit of a religious question, in my opinion. People have a lot of opinions and feelings about that, and both are work well in different scenarios. But zone is where we can really dial it in for sampling for biology, right? Sure. If we know, again, what organism we're really interested in, we can design those zones to really capture that variability, that spatial variability, and make sure we find that organism. So, for example, if we're out hunting for a pathogen, let's say Goss's wilt, because it's something that certainly a lot of folks in Nebraska have been battling for a long time. Mm-hmm. What decision are we going to be enabling? We probably variety selection, rotation, things like that. Tillage, that's obviously a way to combat that. So what information do we need to find from a soil sample that can help drive those decisions? Sure. Should I plant continuous corn? Do I need to, as hybrids become more tolerant to disease, do I need to consider that as a management tactic? And so sure. If we took, let's say, one soil sample per field, we run the risk of not finding the problem, right? But Absolutely. If we say have 2, 3, 4 samples available to us, where can we strategically place those samples to make sure that we find maybe the levels of high inoculum that can help drive the decisions? We're a technology. I think that we're a bit out from really being implemented at a grid scale, let's say as fine as 2.5 acre resolution. Mm-hmm. We've done plenty of fields at that resolution. It's extremely cool to see that data, but we got to get to a point where that's cost effective and really zones a nice way. One last thing along zones, if you don't really have that comfort level and maybe the amount of data needed to create a zone that would be focused on what we're looking for, we have a lot of folks that will sample. Let's take your high yielding part of the field where it's stable, high. It's high in corn, it's high in soybeans, it's high in every crop. Find that lower part, yielding part of the field that's also stable, lower yielding, and then find somewhere in the middle. Give me 3 samples from varying levels of productivity in that field. Let's see what the underlying soil biology is there and see if it's what's causing the problem.
Absolutely. It's a really detailed approach. And I think the best part about it is that you're working with people with what they're already doing, because it makes it really easy for them to see where this fits into their operation right now.
Yeah. There will be a time when what we do is fairly mainstream, and maybe we'll be able to change folks' practices when it comes to soil sampling. But unless we have a really good reason, let's say we're after chasing one specific pathogen or nutrient cycling indicator, as we call the organisms involved in nutrient cycling, and we need a very narrow set of parameters. If we have that sort of situation, we would probably work with an agronomist or grower to really fine tune that plan. But otherwise, we try to align with what they're doing today.
Do you want to ask the next one, or you
Sure.
kind of roll?
Yeah. So some of the applications that you mentioned already are, testing for pathogens, testing for nutrient cycling, microbes. What are some of the potential other applications? Because I know microbes are involved in a lot of processes, such as microbial breakdown of pesticides, pest control as well, such as BT. And then also, what about maybe the environment that the microbes are living in? Is there anything that you're working with to maybe alter the environment based on management practices to favor some microbes or maybe harm others that are negative, like a pathogen?
Yeah, it's a great question, Zach. And really today where Trace Genomics is at is we're really an enabling data set or technology to go down the road that you just mentioned. Now, we don't specifically at Trace Genomics work in those areas where we're actually recommending approaches or even sometimes digging into some of the questions that you've asked, but we work very closely with a lot of folks who do, that ask the same questions that you just asked. How now that we can quantify and perhaps get a baseline of what's going on in soil biology, how can we start to understand that better and to use that to really be better managers, right? And so one of the things we do along that line, we really have a few main areas of customers. Of course, the logical one is we work closely with agronomists and growers, right? Working to drive better management decisions. But another thing we also do, we work a lot with product manufacturers, biological manufacturers, helping them understand two things, how their product impacts the soil microbiome. And also on the other side, how does the soil microbiome impact the efficacy of their products? So two ways you can look at that. Because we have this amazing technology that allows us to really do that. And so that's one of the things that we're really working on. When you start going down the road of some of these really interesting questions about disease suppressive soils and all these really cool things, a lot of that research right now is coming out of academia, and they're really focused on very tiny and very specific questions. And so we also hope to enable that sort of research as well. But really, today where we're at, Zach, is we're really a tool to help enable a lot of that. A lot of our internal R&D is very much collaborative with folks, like as I mentioned, agronomists, with product manufacturers, and really goes to the level of we've done some modeling work looking at pathogens. So if you think about what I mentioned earlier, we quantify the inoculum level. Well, if I recall back to my plant pathology courses, and Zach, you're a DPH student, what do we really got to consider? What are the three things we got to consider?
Well, there's a disease triangle that includes the environment, the host, and the pathogen.
Yeah. So Trace Genomics, we know that we do a pretty good job on one of those. I'm not the world's greatest agronomist, but I can know what the crop is when I walk into a field. So then we have that environmental piece, right? Yeah. And so that's really a big part of it is if you have a high inoculum level, that doesn't mean you have imminent infection and you have yield loss, right? It's not. And so we know that. And we're working on various efforts to model out certain pathogens in certain situations to better understand using our technology what's going on. Now, I also like to think that even this step of quantifying the inoculum level is, I always ask people, what do you do today if you have soybean sudden death syndrome, for example? If you took a soil sample, where would you send that to even get the quantification of the inoculum level? And you can't, there's nowhere to send it. So already that first step is a big step in just being able to quantify the organisms in that soil sample. And we know we can do better by creating models and helping drive very, very specific recommendations, but it takes time.
Sure. Now I think that opens a whole new field of questions for this interview, just thinking about kind of the data aspect and thinking about Trace Genomics as kind of an enabling technology. How exactly are y'all leveraging the data that comes out of individual fields and kind of what you know about that field alongside, and I don't even know, I guess, kind of to start, are you even aggregating data and bringing it all together in kind of a way that machine learning or something along those lines could be leveraged to figure out, okay, these environmental conditions, this pH, this soil temperature is related to this certain inoculum level and actually the presence of disease once we get to, say, R2 in corn? I mean, are those things that you're working towards right now?
Yeah, it's a great question, Jackson. And so that's why I wanted to make a point that we do have a data science group at Trace Genomics, and we're not just a traditional lab in the sense of we're just a data generator. We're a partner all the way through, and we're also an R&D organization at the same time. And so, I mean, our folks in our data science group, they can do things I can't even imagine. Like the end of my statistical ability ends with the last A in ANOVA, right? I mean, I'm done after that. And they're using these really fancy machine learning techniques and applying that to really look at our data set and try to understand what's going on. Because if you think about every soil sample that we capture, if we're going to extract the DNA of the fungi and bacteria and sequence that, that's a massive amount of data right there. And then you add in, the chemical parameters, and we do a very comprehensive panel of chemical parameters of the soil. And then you start adding other layers, productivity, whether it's yield or imagery, as applied layers, you name it, and it becomes a fairly hefty data set that really needs some good statistical techniques to tackle. Now, what do we do today from the customer standpoint that you get with every sample? We do aggregate the data anonymously at the national level. And what we do is what we call benchmarking. And basically, we'll take all that data, let's say for corn or soybean, so that when samples come to us, the folks collecting the samples have identify the past crop and the current crop. They identify if they're sampling in season or out of season. They typically were supplied the GPS coordinates of the center, probably, of a composite sample, and we have all this information. So what we might do is take all the corn samples and we'll aggregate that data and we'll create benchmarks. And so what the benchmarks are is, as Zach mentioned, we have good IPM principles such as the disease triangle, but we also have economic thresholds, economic injury levels, and things like that. And currently, we're always working on capturing that data. But until that time, why don't we tell folks where your samples lay in relation to everybody else's, right?
Sure.
So what we do is actually we fix a benchmark that you can compare the data from your samples to all of the corn samples that we've received. Now, to take that to another level, we work with folks from the Gulf of Mexico all the way up into Canada, and let's say they're soybean producers. So here I am in Arkansas one day talking to a soybean producer. Then I'm in Southern Illinois, I'm in Iowa, I'm in North Dakota, and I'm comparing the amount of soybean sudden death syndrome or phytophthora or white mold from one day to the next across those geographies. The folks in Arkansas are going to say, other than the fact that we're both growing soybeans, there's a lot of difference to North Dakota to Arkansas, right? So why are you comparing my pathogen levels to theirs? And so what we've done over time, as we've really collected a lot of samples from a wide variety of areas, is we've created what we call smart benchmarks. So smart benchmarks are still an aggregation of data across numerous farms and growers. But what we're doing is aggregating at the level of like soil properties, soil parent material, environment, and cropping systems. So you could say, well, you're not necessarily comparing to the field next year's, but you're comparing it apples to apples.
Right.
I'm comparing all the soil samples that are collected in Southeast North Dakota, or I'm comparing all the soil samples that are collected in Northeast Arkansas, but I'm not comparing the two. And so, that was one of the lessons I learned a long time ago when I was in the seed industry. We talk about this idea of yield stability and hybrids and varieties, right? This hybrid did great across 5 states, and it just kicked everything, and inevitably the grower would say, well, I really want plot results from the nearest plot, in the county or whatever. I don't care if this hybrid did well over 5 states in 3 years. I want to know what it did, next to my farm, right?
Yeah.
So it's no different than pathogens. You don't want to compare what's going on in Northeast Arkansas to what's going on in North Dakota.
Sure. I think that makes a lot of sense. I mean, it's the smart benchmarking it's just kind of a crazy concept. I know farmers really like to get a lot of feedback. I mean, have you had any farmers that ask you if the data can be used negatively against them? I'm going to play devil's advocate and say, like, in this data security, place that we are in digital agriculture, are farmers worried that, okay, if I know that I have this disease, is this going to be something that, crop insurance or somebody else, my banker, is going to know about, and it could potentially adversely affect me? I don't know. How secure is that data, I guess, that Trace Genomics is generating?
So sort of a similar scenario to if I send my DNA off to 23andMe and then next day they give it to my health insurance company,
Yeah.
and I'm uninsured,
Exactly.
it's sort of the
Yeah.
same thing.
Yeah.
Yeah, I mean, that's a very viable concern, but we have a lot of folks in the company that have spent a lot of time in ag tech and have really been very observant of that. And so
Sure.
guidelines for sharing and for the technology are very strict in that we don't share any data. We aggregate anonymously, obviously, and some certain things like that. So we're not sharing. Being a startup, people worry, well, okay, today you're a startup and you say, okay, we're not sharing your data. But what happens if a large company or somebody buys you out? Then all of a sudden they have your data.
Yeah.
And certainly that's a concern as well. And so that's a great point, Jackson. And we do our best to keep people's data safe, and we don't share it with anyone.
Sure. And I think that's the right standpoint. It's just something we always, I don't know, I feel like we ask a lot when people are collecting a lot of data, because it is such a big deal, and something I hear from a lot of farmers. They're concerned about in this new digital age that we're in.
Yeah, well, I mean, this is probably something you've covered on other podcasts with people that are much more astute than I am in this area. But who paid for the data? Who literally gave you the money? Then that's that person's data, right? There's a lot of nuances to that, but I believe that that's really the way it is. I'm a farmer, and I've worked with a lot of different programs and different platforms, and I pay for that service. That's my data, and I don't want it to be sold to anybody.
Mm-hmm.
Now, am I naive enough to think that those companies aren't using it to learn more, to create aggregated anonymous insights? Sure, but for them to sell it off, that's not something that a lot of growers would be excited about.
Absolutely. Yeah. So kind of just keeping on moving on this kind of data aspect, you mentioned earlier that you're, at least at Trace Genomics, you're trying to incorporate some other data streams into better modeling what's going on out there in the field. And you talked about as applied data, you talked about yield data, you mentioned imagery. What exactly does that look like, and how exactly are you able to incorporate all those different data sets? Is it through partnerships? Is it, are these mostly just done on kind of a research scale where you have full control over the farm, or is this something that is really at commercial scale for everybody that works with Trace Genomics?
Yeah, it's not a commercial scale. A lot of the work that we do where we incorporate productivity, whether it's imagery or yield monitor data, where we might bring in as applied, a lot of that is really in an R&D phase at this point in time. We do have a really fantastic customer portal where folks can create orders to submit their samples, where they can geospatially see the data, where you can manipulate the data. Folks can also get a export a CSV or a Modus XML file. Trace Genomics, we're really excited about something we did last fall, which is we added a majority of our biological parameters to the Modus standard. And so that's pretty exciting. But certainly one of the things that we're not doing today is allowing folks to bring in those layers and to do any analytics today in our customer portal. Sure. It's certainly something that would be really exciting for the future. And there's so many different file formats and issues with how clean data is and all that we have to overcome. And that's why it's really relegated to really an ad hoc basis and more R&D today. But there's no doubt that we'd love to work with people who have really good data and to really drive those deeper insights. Because as Zach mentioned, that disease triangle, when you get to talking about the pathogen, that's you got to have more than perhaps just the quantity of the inoculum. You got to have some of that other data. And that brings up another point, weather data too. I mean, that's something that we are always working on, incorporating good weather data and using that to really further this along.
Sure. So you mentioned the platform and kind of the customer portal and your answer there. And I guess I'm just wondering, I haven't actually seen it myself. What does it look like for a customer who's receiving a Trace Genomics report? What are they able to go in and see? What do those recommendations or measurements look like to them, and how actionable is that really for them in the field?
Yeah, so today, if you collect a soil sample, We'll enter, as I mentioned, we ask for a bit more information than your standard soil testing lab would. We want to know a bit about the cropping history. We want to know the depth of the soil sample, the data was collected, also the GPS coordinates for that soil sample. And then you're able to upload a shapefile boundary and also zone boundaries if you have them into our customer portal. And when the results are ready, whether it's the quantification of pythium or the quantification of nitrification or pH, all that information is geospatially rendered in our online customer portal. And so people are able to manipulate that. We do a bit of sorting and surfacing of issues there as well. For example, for a pathogen, our quantification of pathogens, that inoculum level is in parts per billion. So folks that are used to traditional soil testing, you might get your Bray phosphorus in parts per million or your potassium value. When we report our pathogens, we report those as an absolute quantification in parts per billion. And so you can see geospatially rendered that map. And then we also do a bit of rolling up to the field level to be able to like I mentioned, surface issues. Like, okay, you have a field that has really a hot spot for GOS as well. Okay, you need to take a look at this first. Versus fields where we have less inoculum where you may not need to focus on it right away. All that data is available all the time. It's just that there's a lot of parameters we want people to sort of help. We want to help folks sort through that. As I mentioned, we have a lot of ways to get the data out, whether it's a PDF report or a CSV file, if folks want to take that data and put it into their favorite FMIS. But we also have a view where you can do some sorting and filtering. It's our legacy portal, and it's a bar chart. You're able to sort by farms and fields and different parameters in order to manipulate the data. Now, one thing you asked about were recommendations, and we don't produce any recommendations today. The closest thing would be the benchmarks that we talked about earlier. Creating fertility recs or creating variable prescriptions and things like that is not something we do. We're happy to work with folks. We do have APIs with a few of the major softwares that folks can grab our data and run them into those programs and to create recs, but we don't do that today. Obviously, it's something that we're extremely interested in, and we're working on it. One thing that we're working on right now is combining the traditional chemistry information that we get for phosphorus, but also combining that with the biological information we get from phosphorus, and really putting those two together to help drive better phosphorus management decisions, but also to help drive decisions when it comes to biological products that impact phosphorus and phosphorus cycling. And so that's probably going to get us a bit closer to the recommendations, and really working with folks to align that with various tools that they have, whether it's phosphorus fertilizers, enhanced phosphorus fertilizers, biologicals, and things like that.
Sure. I think it's valuable that you're kind of waiting to get into the actual recommendation space, because if you have too many wrong recommendations early on, I feel like it kind of ruins the trust factor. And I'm not saying that they would be wrong, but it's one of those things that you definitely want to get right for a farmer.
Yeah, and we cover a fair bit of the United States, from apples and lettuce to corn, soybeans, cotton. We really rely on that local expertise to take it that last mile, but we'll work with those folks. Mm-hmm. We may not write recs, but we work with folks who do and enable them to use our information to really drive those decisions.
That's awesome.
And so it's not that we don't want to do that and that we couldn't. I mean, we have a really beautiful platform that allows us to do that. It's just that we really want to put the data in the hands of the experts and help them go that last bit.
I think that's really valuable. Kind of on that same front, have you heard any stories or been part of any stories that are particularly successful outcomes that growers have had out there from using what Trace Genomics offers to them?
Yeah, there's a couple of ones that get me really excited. And a while back, we were working closely with a grower who had basically more or less quit growing soybeans, had a terrible time with soybean sudden death syndrome, and just was going to sort of wait it out and mostly just focus on corn. And we were able to sample a field that had been in corn for quite some time, and to really get an idea of what the inoculum level was at that point in time in that field for soybean sudden death syndrome. And we were able to get some soil samples, compare some fields that had been rotated corn and soybeans every year and that sort of had known levels of SDS, and really give the grower some peace of mind that the levels had been reduced in that field, and that perhaps it was okay to reincorporate soybeans into that rotation. We all know as good agronomists that continuously growing a single crop can certainly be done, and there are no laws against it, but it's certainly, every once in a while you got to break that rotation and add something back in. There's another example that got me thinking about, and is a few years ago, I was working with a grower, actually agronomist, excuse me, and we were going over the results for the first time, and I was really trying to help the agronomist understand how to interpret our results. So I went through a few fields and interpreted the results, and I pulled up, at the time it was still bar charts, but pulled up a bar chart of another field, and it happened to be charcoal rot in soybeans. And I said, looking at the data from this field, what would be your interpretation? And the agronomist directly looked at one of the quantifications, which was in the thousands of percents of our benchmark value. So it was pretty high value for charcoal rot, and proceeded to pull out his phone and showed me a picture of a soybean plant that had some extreme charcoal rot issues. And it was exactly in that area where we had found really high levels of inoculum. And so that was really exciting. And of course, then people are really buying into your technology and what you're doing when you can really confirm their knowledge of the field and what they've seen.
Yeah, it really makes you feel good when a farmer can say, yeah, that's exactly what I've been seeing, point to it on the map. That's my problem. And
yeah,
it just makes it so much more tangible.
I know you're the one asking the questions, but I might skip to one of your questions. You asked about how can people perform trials to test the efficacy or the validity of what we do. And I tell you what, people do that every day. And I think it's great. I mean, we want to be as open and transparent with folks about our technology as we can to build that level of trust. And so we often get blind samples, ABC 1, 2, 3, and folks send them in, and they know this came from a level area of the field with high infestation or low infestation or whatever, and they send them to us to see if it tracks. I mean, I was just talking with one of our scientists today on some development work. We're working closely with a group of agronomists, and they want to test our capabilities out by sending us samples, and they're not going to tell us is it high or low or in between to see. I've even had some agronomists, and I love this. I have no problem with this. They name samples high, medium, and low, and they send them to us. And we get results back, and I'm just sitting there going, oh, well, high, medium, and low. The results are, completely the opposite of what I'd see. And I'm a scientist. Scientific integrity is of utmost importance to me. The data is what the data is, and I believe in our process. So we take the data to the customer, and they go, we go, it's reverse of what high, medium, and low labels, and they go, oh yeah, don't worry about that. We flip the labels.
That's pretty awesome.
It's interesting. Now, you don't expect that every time. Most people don't do that, but I've had a few instances where they've done that, and I see, test us out. I mean, we're, if you think about the technology adoption curve, we're really in the innovators, the early adopters. We're talking with the folks that are trying all the new technologies and want to try it out. And typically, they really enjoy these new technologies. They have a hefty skepticism, but they also give you a lot of room to run.
Sure.
They know you're not perfect. By the time you get to the average grower, the average agronomist, you got to be bulletproof, right? You got to be a technology that's really tried and proven. But we work a lot with the innovators, and it's a lot of fun to see how we match up to their expectations.
those people help you get there. They're almost like partners in the development when they push you like that and make sure that everything is, I guess, performing up to their standards in a way. Yeah, absolutely. So I guess, we're kind of running short on time here, and I want to be respectful of your time. As I kind of like to move to the last couple of questions. And number one, where can our listeners go to learn more about Trace Genomics? I mean, obviously there's a website, but are there other resources you would point them to?
Yeah, certainly the website is a great place to start. And actually that through that, you can filter to our sales folks or our customer service people. I'm always happy to answer any questions that folks have. We also have a few sales folks out in the field, but a lot of the work that we're doing, as I mentioned, is really going through agronomists, through retailers, and I'd be happy to help folks find their nearest retailer that they may be able to buy our testing through, and that's probably the easiest way to get a hold of us.
Awesome. And I guess the last question that we always ask in these interviews is for some advice. And I guess, what is some advice that you might have for individuals that are trying to stay ahead of disease and other problems that might be occurring in their field and looking for that way to get their next edge over their problems they're facing in their crops?
Sure. Yeah, we're always everyone's always trying to do a better job, whether it's financially or agronomically, environmentally. And so, these tools like what we have at Trace Genomics, to be able to take that first step, as I mentioned earlier, if I want to send in a sample today to get quantified for soybean sudden death syndrome, you can't send it anymore, or GOS's will, right? So we're enabling that capability and that first step, but it's got a long way to go. And here's something interesting to think about. If we're geospatially rendering the amount of Pythium or Phytophthora in your field, think about the different management tactics that we've just enabled. I think we just enabled you to be able to place varieties for, let's say, Phytophthora and soybeans in a field. You may be able to place a variety that has low Phytophthora tolerance in one area, and in areas where we find a lot of Phytophthora, you may want to put a resistant variety. Same with Pythium. So, for example, seed treatments are really have gained a lot of popularity, not just in soybeans, but also corn. You start to think about, if you geospatially knew where there were differing amount of Pythium in that field, and you could have the ability to potentially variable where you put different seed treatments. Yeah. Now, what I would say is our technology is a bit out front of people's appetite, maybe ability to do those sort of things.
Sure.
But if you look at ag tech and some of these things, it's a back and forth, right? We have equipment gets out in front of the data, then the data gets out in front of the equipment. And so it's very interesting that we have a technology. If you were able to soil sample, you could actually enable some of these decisions that maybe people aren't ready to enact yet. So I think that's really cool, because seed treatments on one hand have been pretty cost effective, and there's pretty decent ROI there. But I think maybe one of the things will be the environmental aspects of some of these things. It won't be necessarily that we're enabling folks to really save large amounts of money, but maybe to be a little bit more environmentally conscious where they're doing some of these tactics.
Sure. I think that's super interesting, especially about the interplay between the machinery and kind of these decisions and recommendations. Because I remember an episode we had about multi-hybrid planters, and it seemed like those never seemed to really catch on in many places, and maybe aren't even really being produced by some companies anymore. But if you think about what you just talked about, where you can have, treated versus untreated seed, and put those in the right places, maybe you've got two different hybrids that respond differently to different disease. I mean, all of a sudden that data that you've now provided makes that multi-hybrid planter worth having, so.
Yeah. One thing we haven't talked a lot about is our nutrient cycling indicators. And you think about if I knew where there were higher or lower amounts of nitrifying organisms, what sort of decisions would that drive? Obviously our blanket sort of decision making, right now for, let's say, fall anhydrous ammonia applications, let's just wait till the soil temperatures cool down. well, okay, when should I be putting on a nitrification inhibitor? I have those two sort of things at my disposal. I can wait till the soil cools down, I can put a nitrification inhibitor. What if I add a layer of data that says you don't have very many nitrifying organisms in this part of the field, or you have a high amount? Can we make better decisions along the lines of nitrogen management, and specifically, looking at nitrogen loss?
Absolutely. No, that's super valuable. We do fertigation research right now. We have a lot of different ways that we try to zone fields out and figure out where to sample. I can just imagine having that indicator would be probably the best layer you could possibly have, because what we're dealing with is mineralization and kind of that availability of nitrogen that changes over time. So if you had that layer, it's like you suddenly know exactly where to be looking in your images to evaluate where that nitrogen should be applied. So
yeah, we also quantify the quantity of denitrifying organisms as well. And so that's and we have one other quantification of nitrogen cycling, and that's really a process that's the reverse of nitrification. So it's nitrate back to ammonium. So we start to get a really holistic picture. And then of the chemical parameters that we quantify in every sample, we quantify nitrate and ammonium. So when it comes to nitrogen, there are actually 5 different, 2 chemical and 3 biological parameters that we quantify in every sample, to really help people get a better view than they've really ever had before from a soil analysis standpoint.
that's awesome. I kind of wish we would have had John during our nitrogen management technologies series as well here recently. Maybe we get back around to that topic. We should have you on again to talk about that side. So
I'm happy to come back anytime and talk about any specific topic you want. We're doing a fair amount of research in that area. Now, one thing that I would never want to allude to anybody is that we're really setting out to be the 500th nitrogen model, 499th nitrogen model. And there's a lot of really great companies spending a lot of money and effort on that. We know our part of the world is really the soil quantification and helping that. Maybe we can inform model creation and things like that. We know we can help agronomists and growers at least get a baseline of what's out there and to be able to understand what they're up against. But we're really love to collaborate with those people and really to help better understand nitrogen. It's a tricky thing, and it's a big nutrient that has a lot of not only agronomic but as well and financial and environmental impact. So
absolutely, yeah, it's a wicked problem.
Yeah, well, it keeps us agronomists in business. It's job security.
Awesome. Yeah, at some point it would be nice to take a look. I know it seems like a lot of these soil biology solutions are going to be really big pieces of management here in the next few years, start to become a lot more prevalent. So it'd be great to get a look at it.
Yeah, we're really at the beginning of this. Like I said, there are only a handful of companies that commercially make this technology commercially available. And so we're really enabling folks to start looking at this. One interesting thing, this is a bit anecdotal, but when we take a soil sample and we extract the DNA of the fungi and bacteria from that soil sample and we run it against our bioinformatics database. So it's got all the genome assemblies and everything that we check for. Typically, we're only able to classify about 10% of the DNA in the soil sample. So that means 90% of the DNA, just the fungi and bacteria that we extract from a soil sample, at this point, maybe is not unknown, but it hasn't been sequenced and genome has been made available. So there's a tremendous amount we still don't know. Now, interestingly enough, a lot of the agriculturally important pathogens that we deal with on a daily basis have been sequenced. A lot of the nutrient cycling indicators that I talk about, those organisms have been sequenced. The genes are known that confer that ability. There's a lot that we know, but think about that: 90% of the organisms in that soil sample, for at the very least, haven't been sequenced and probably haven't been cultured and identified. So, and they're doing something. They wouldn't be there. I've taken enough biology and ecology to know that they're serving some function. And they're probably interacting with all those organisms that we are interested in. They're probably interacting with all those biological products that we put on and in furrow or soil applied. They're probably interacting with the seed treatments, fungicide seed treatments that we're putting out there. So the more we start to know about these, be able to very least know and quantify these organisms, the more we'll be able to start learning about the soil and all the interactions. But well, I tell you, we're at there's been a lot of soil microbiologists that have been working diligently for a long time, probably over 100+ years, but using molecular techniques in soil microbiology at this scale is a really new thing.
Yeah.
We're going to keep trying to figure out better ways to do it. Hopefully I gave you a good overview of what we're doing. If you're ever or anybody's interested in looking at our customer portal, I'm always happy to run folks through that and answer any of the questions I can.
Thank you very much, Dr. Erik Christian of Trace Genomics, for taking the time to join us on the FarmBits podcast. I really enjoyed getting to hear some of his expertise that's been gathered over many years spent in the industry with a lot of different facets of crop production. One of the things that I thought was super interesting about the episode is number one, how they're leveraging data to basically create better insights for farmers through anonymized data, and they're also providing like these specific benchmarks for farmers. But really, I thought it was great when he got to the very end of the episode, exactly how some of these insights can be turned into management decisions to help make better use of the precision equipment that we have out in the field.
Yes, definitely. And a lot of the agronomists, growers, and other companies that Erik is working with are definitely early adopters of this technology, and they're driving agriculture forward into the future.
Absolutely. Yeah, there's a lot of cool stuff. I'm sure we'll see a lot of cool stuff from Trace Genomics here in the next few years. We hope that you enjoyed that interview as much as we did, and we look forward to you joining us next time here on the FarmBits podcast. Thanks.
Thank you for taking the time to join us today on the FarmBits podcast. If you enjoyed this episode, please subscribe to the podcast on Spotify, Apple Podcasts, YouTube, or wherever you listen to podcasts to be informed about the latest content each week.
We welcome your feedback, so if you have comments or questions for us, please reach out to us over email, on Twitter, or in the review section of your favorite podcast platform. Our contact information can be found in the show notes.
We'd like to thank Nebraska Extension for their support of this podcast and their commitment to providing high-quality informational material to the members of the agricultural community in Nebraska and beyond.
The opinions expressed by the hosts and guests on this podcast are solely their own and do not reflect reviews of Nebraska Extension or the University of Nebraska-Lincoln.
We look forward to you joining us next week for another episode of FarmBits.